Chidima, how are you? How are things? How's been back at school? I'm good, thank you. I am good. It's been a busy day. Busy day, working away. So I thought I would jump on here. And whilst my video is downloaded from my group tuition, I went in today even though it was, inset day for your teachers and you went in. What were you doing? Hi. Wait, did you make the bio exam prep thingy for summer 2025? Yes, I did. I did the videos on my YouTube channel. Yeah, that was me. That was me. Thank you for the likes. You were training them. Oh, cool. What was your project on? That's really cool. I don't know if you remember, but it was the competition that Clash. Oh, yes. No, I do remember. Hi. How are you? I do remember. Thank you so much. I was trying to find your channel but couldn't find it. Your predictions were so. Oh, thank you. Do you have any tips on the beetroot practical? I have mine tomorrow. Are you AQA? When you say you've got one on the beetroot, is it practical 4 you're talking about? I am good, thank you. I am good. Yeah, it is AQA. So, with regards to the beetroot practical is actually quite a nice one because all you're going to have to do is cut up some beetroot. So, if we think about this, if we have our beetroot, we're going to have to take, now it depends how you're going to take the section out of the beetroot. So you can either do it with a cork borer, which is quite an easy piece of equipment to use, or you can just use a knife and you're going to cut your beetroot up so that you have the right sized material that you are looking for. So you've got to think about surface areas. If I've got my beetroot and I cut out a section from my beetroot, I'm going to want to make sure that my surface area is the right size because I don't want to expose too much or too little. I want to try and keep it as accurate as I can, and therefore I've got my control variables. So it's really important that you bring in what are you changing. So with regards to your practical, is it going to be temperature changing or are you changing the solvent concentration? Temperature. So if you're looking at your independent variable, therefore being temperature, so make sure you think of 5 values at least. So I would probably, ooh, my tripod is broken, so I've been trying to get it. Is it going to actually stay up? I need to buy a new tripod. Oh no, you're just going to fall, aren't you? Ooh, crumbs. Let's hope that doesn't fall again. If it does, I'll catch you. I'll catch you if you fall. So temperature. Answer your question. What's the difference between GCSE and A level? GCSE is it's kind of like the building blocks to start you off. It gets you interested in the topic area, and then the A level really takes you to that nitty gritty part of the content. What's my favorite organelle? My favorite organelle, it really just depends because there's so many different ones, but I love the mitochondrion and I love the chloroplast because there's so much detail to go into with that. Right, temperature, pick 5 values normally, but I would go anything from 10 degrees C up to about 60 degrees C. So I'll probably give you 6 temperatures. So whenever you're talking about the independent variable, you want to give them that information. So I'm going to go from 10, 20, 30, 40, 50, 60. I want a nice range because I want to make sure that I have enough to see a difference. And I want to get up to a point where I have denatured my proteins enough so that the phospholipid bilayer isn't intact. I need that pigment to leak out. Because the key thing to note whenever you're doing the beetroot is you're thinking about that phospholipid bilayer. So the phospholipid bilayer is the one that's going to maintain that pigment inside. So the fact that I have my phospholipids, and this is my bilayer of my phospholipids, what I want to make sure I'm doing is I'm actually denaturing, breaking apart that membrane, because there's an increase in temperature. If there's an increase in temperature, that's going to result in an increase in kinetic energy. I was actually doing this with my student tonight who is AQA. So somebody did ask, do I do AQA? Yes, I do. I tutor both AQA and OCR. I made my resources OCR because that's what I was teaching in the school that I'd gone into support. And then I also realized there was a lot of AQA out there already, but there was nothing for OCR. But I do actually tutor both. Anyway, back to this. Increase in temperature means increase in kinetic energy. If there's an increase in kinetic energy, you're going to have more leakage. And what you tend to find, so you're going to maintain your surface area. So that's a control variable. So my surface area will be controlled because I might do 2 centimeters by 1 centimeter. I obviously need to wipe it. I want to wipe excess away because I want to ensure that my concentration that I'm looking at, how much of it is actually being released, is based on the actual membrane being disrupted. That's what, wow, 1,000. Thank you so much, guys. Sorry, I'm just currently looking. Hi, Maeve. Do you need to know more about prosthetic groups, cofactors, and that in enzymes in a lot of detail? No, you need to know about the heme group for hemoglobin. So you need to know that that is a conjugated protein. The chloride ions which are needed for amylase is your cofactor that you need to link into that. But apart from that, no, there's not actually that much you need to know. If they're going to give you anything, it will be that they ask you to decipher the information that they've given you in the question. You have a test on hormones tomorrow. Can you just go through the key concepts you should know, please? Well, to answer your question, oh, hold on one second. Does that help you with your question on the practical when it comes to the beetroot? Basically, keep the surface area the same, keep everything else the same, so that the only thing you're changing is the temperature. You're going to use a water bath for that temperature. You're probably going to use a colorimeter. If you've never used one of them before, don't worry too much. The whole point of a colorimeter is that you are looking at the light going through. If the pigment has leaked out into the solution that's surrounding your sample. So if I take my beetroot and I place that into a beaker of water, now inside the beetroot there should be the colour. I'm going to colour it pink here, and obviously my liquid which is surrounding it is going to be clear. In this case it's blue. What will happen is, if the pigment is leaking out because the phospholipid bilayer is no longer intact, what you're going to do is you're going to sample that and you're going to shine a light, and that light will go through a filter, the filter that is appropriate for the beetroot pigment. So it's probably actually going to be a blue filter that you use. And then you're going to have that pass through your cuvette, which has your sample in it. And you're going to look at the absorbance, and what you should find is as you increase temperature, it will do that, and then it will plateau off. So you'll have your starting point, and that is an intact membrane. As the temperature increases, the membrane becomes less intact, more permeable. Because it's becoming more permeable, that's going to result in more leakage until you get to a point where all the pigment has been released. Which is there. And guys, if you can't see, I am actually on my channel with a board displaying this. Right, let me see where I got to. Where did I get to? Is this similar to Edexcel? Yes, there's a lot of overlap in the biology content. It's just important you make sure you're looking at your specification. Is there a difference in transmission and absorbency? Yes, there is a difference in the reading that you get. So if you're getting a reading which is the transmission, it's looking at how much is going through and therefore being transmitted through the sample in your curvette. If you're looking at absorbency, you're talking about how much pigment is absorbing the light and therefore not allowing it to transmit through. So it's about the reading that you're doing on the colorimeter. Your biomarker that determines your predicted grade for the research is next Wednesday, maybe. Okay. Good luck on that one. So next Wednesday, I'm going to try and go live on Wednesday as I always do. However, I might not be able to because I've got to look after the girls. So if I'm not able to go live, it's kind of why I've gone live tonight. If I'm not able to go live, one, I need to buy a new tripod so I can do it, but I'll try and do it from the house to help, because that will mean you can have another one. You are very welcome. Right, what was application is the hard part. Yeah, you know, application is hard, but if you take the time to go through questions, hormonal content, I am coming, I'm trying to find your question, but I am going to do hormonal content. But what I would say is the application is about reading the question. It's about figuring out what is being asked. Where's the topic area that they are trying to assess you on? If you know your content, you then need to think, right, what have they told me in this question? Have they said it's enzymes? Have they said it's biodiversity? Have they said it is classification? What is it they're assessing you on, and use that. How often do I upload a new OCR A-level biology? To be honest, at the moment, I'm not really uploading that many new videos. I need to finish off my transport and plant study guide. That's the first thing I'm going to be doing. And then once that's done, I will start making more videos again. I have to prioritize my income that comes from the people who I do one-to-ones and group tuitions with. So they take priority. And when I have some spare time, then I put the rest of the content out there. The content, the whole content is on the on my YouTube channel, so you do have access to all that. What I would say is that the stuff that I'm making just now with regards to my study guides, that's me doing it in a way that it makes it make more sense. So I have been prioritizing them, but I am also just about to throw out some new videos for GCSE. If you bought the group sessions for OCR, would it be the same for AQA? I want to say actually no. There is a lot of overlap, so but the style of questions. So I tutor quite a lot of private AQA students, and the questions are different, although I kind of show AQA to OCR students and I show OCR to AQA students. There is a lot of overlap, but the problem I've got is if you were to attend the group tuition sessions, it's aimed at the OCR side of things because they are, I know that content through and through, in and out, so I can jump all over the place. Whereas with AQA, although I know the content inside, I would have to be double checking it against what I've already planned. So I don't want to say come along. I mean, please feel free if you want to come along and see what one's like. I'm not able to run it this week because I've got to be with the girls, but I wouldn't want to give you any false sense of information here, if that makes sense. I'm considering offering AQA group sessions. I'm not sure if I will or not yet. The problem is not that many people want the group sessions, and so I have to prioritize where the money comes in from, obviously. Cholesterol regulates fluidity, but whether it decreases or increases depending on the temperature. Yes, so if it gets too low with regards to temperature, what you have to be aware of is that you don't want the membrane to not move at all. So if the membrane doesn't move, then nothing's going to get through, and that's a problem for the cell. If the membrane becomes too fluid, too many things can get through that can affect the function of the cell. So you want it to be there to regulate the fluidity of the membrane. So not make it too low, but also not make it too high. Right, okay. Hormonal system. It actually links to the membrane, so I will link it to what you were just asking about. Excuse me, with the membranes, but I'm going to go on the hormonal side, which is module 5, because you, if you're still here, I apologize if I've talked too long and you've disappeared, but the hormonal system, it's normally on a Wednesday, Norva. Hi, welcome. It's normally on a Wednesday, but I'm doing it tonight because I might not be able to run it this Wednesday. I'm going to try, but in case I can't run it on Wednesday, then I jumped on here tonight for that. What am I doing? I'm trying to find the hormonal content, but I seem to have done it in a completely different place. This is where I'm like, where have I actually put the stuff I want to show you? And right now, because I'm trying to find it really quickly, I can't find it. I might just have to go slightly different. That's fine. Right, let's start with histology. Maybe you're still in here, because I think it was you who said you've got your hormonal thing, or did you say you had your test? Who was it? Was it you who said you had the hormonal side of things? I'm trying to find it. How would you explain to any level class joining of nucleotides forming polynucleotides as clear? Zingy, give me a second. Let me finish doing what I was doing on the hormonal system, and then I will come back to answer that question. Right. Let's get rid of that. And then we're going to go hormonal system. If we're talking about hormonal system, we're probably looking at glucose concentration. One of the things to note is about how the cells communicate with them. So let's go. I'm going too far now. Right, here we go. So blood glucose concentration. If we're looking at hormones, we tend to go down the route of talking about your blood glucose concentration. So this is homeostasis, and also linking into the way in which the body communicates. So you have cells that will release different hormones. Those hormones, remember, are produced through the process of exocytosis and secreted into the bloodstream. So when we look at blood glucose concentration and we look at the homeostasis part of that, what we're talking about is maintaining a level which is kept within a very set limit. So your blood glucose concentration should be between 4 and 8 millimoles per litre of blood. So we monitor that and we regulate it through the use of the pancreas. So 1 glucose is the respiratory substrate of choice. So the glucose is broken down within the cytoplasm through the process of glycolysis, and therefore we want the glucose to be available to be broken down. Now, some cells will store that glucose as glycogen, so we will need to break that glycogen down to release the glucose, or we need to metabolize the fat that we have. So ideally, we want the blood sugar to be at a level that means respiration can continue, and then we need to signal if the blood sugar levels get too low or if the blood sugar levels get too high. So we want to either store it if it's too high, or we want to utilize it if it's too high, or we want to increase the level by secreting a different hormone. So respiration is one of the key reasons why we want to maintain our blood sugar levels. Obviously, if we have got glucose in the bloodstream, glucose is a polar molecule that will bind to the, I say bind, it will form hydrogen bonds with water molecules because it is able to dissolve. It is a soluble substance, it can dissolve, and because of that, that is going to affect the osmotic potential. So if you have too much sugar in your bloodstream, that will cause water to leave cells and go into your blood vessels. That will cause the volume to increase. That's a problem. If you have too much water, that will also have an effect and therefore could cause cells to swell up and to burst. That becomes an issue. So it's important that we understand that our blood sugar levels, we want to maintain them within this set level. And to do that, we are going to need to monitor it and regulate it. So because of respiration, because it is the respiratory substrate of choice, osmosis to keep our cells functioning, to keep that level within all the cells so that chemical reactions can occur. And so therefore we need to understand that the pancreas is the place where it is going to be monitored and regulated by. If we had too much water, yes, absolutely. If the cells take on too much water by osmosis, that could cause them to burst, and we don't have a cell wall to stop that, to get our cells to that turgid point. So therefore, if they're taking on too much water, that will keep expanding until there's too much there that will then burst. That phospholipid bilayer can only create a barrier if it is maintained and structurally sound. And so therefore, yes, if there's too much water, we are going to result in our cells swelling up and bursting. But our kidneys are involved in, and our hypothalamus is monitoring our water levels constantly. And so because of that, we should be excreting. So I'm getting a dry mouth because I'm talking very fast, talking a lot. And so I have my water where I'm going to take a drink, and that is therefore going to hydrate me to keep those levels up. However, if I drink far too much too quickly, I'm going to need to run to the toilet to excrete that excess water. Do my videos cover all the content? Yes, they do for OCR-A. Absolutely, they do. So right the way through, it is all the way from module 2 to module 6. Zingy, I haven't forgotten about you. I'm going to cover this and then I'll come back to you with the nucleotides and forming polynucleotides. But if you want something specific, Zingy, you want to look at 2.1.3, I want to say, nucleic acids and nucleotides playlist. The practicals user, are you AQA or you OCR? Because OCR don't have set practicals. They have got the PAGs, which is PAGs 1 to 12. I'll come back. OCR-A. So yeah, the practicals are dependent on PAG 1 right the way through. But with regards to the excuse me, practicals that are written throughout the specification, yes, my videos do cover them and talk through them. They don't show the practicals because I've done them at home. However, they do help. Why is postpone? Siggy, he's not, he's OCR, he's asking some questions. I will answer you too, as well as doing this. Be patient. Right, pancreas, exocrine, you have your regions within your pancreatic duct. You also have the islets of Langerhans. So one of the things they love to throw at you is histology. So your islets of Langerhans are little islands. And so therefore you also have the fine branches of the pancreatic duct. So when we talk about enzymes, so if you're AQA, you should know about your exocrine and your endocrine glands. So your endocrine glands are the ones that are going to secrete those hormones into your bloodstream. Your exocrine gland, i.e. the pancreatic duct, they are secreting the enzymes which are going to go to your intestines. And so therefore you should know about both. With OCR, they are more looking at your islets of Langerhans and the fact that they're split into the beta cells, which are going to secrete insulin, and your alpha cells, which are going to secrete glucagon. So one of the things I would say to you is make sure that you are able to identify where your islets of Langerhans are. Now I can see the islets of Langerhans are very different to my acinar cells, which are the ones that surround it, and that's through the use of staining. So when you look at staining, differential staining, using different stains will allow for us to identify our different regions. So that's why histology is really important. So you can see that this one here is my islets of Langerhans. You can see really small islets of Langerhans in the diagram, and we've zoomed in. So we've used a higher magnification. So we've changed our objective lens to be able to observe the different features. So one of the things they can do in the exam is, yep, they are your light purple ones, give you a picture like this and then ask you, after giving you some information, why they might stay in a different color. Why is it the nucleus is darker than the cytoplasm in these cells? Why is it that you can see? So it's about different concentrations of, for example, proteins that are being produced. So it's important that you're looking at both. It's important to do both, by the way, just to be clear. If you're doing revision, watching videos will only get you so far. Doing past papers will only get you so far. It's about doing both. It's about using what you can, as many resources as possible, to expose yourself to the types of questions they'll ask you. But also it's about practicing your written ability of answering those questions. You need to make sure that you are also writing down your answers, not just memorizing mark schemes. I am always surprised how many students think they can get away with memorizing mark schemes, and that won't help you. Also, using mark schemes to formulate your right answers is going to help you. If you take a mark scheme and then you convert that into your own written answer, that will help you more than if you are just going to memorize and write exactly what they've said, because the questions will never be the same. The mark schemes will never be the same. Every year, the mark scheme is specific to that year of those students, that cohort who sat it. So they accepted or did not allow certain answers based on what was actually given in that year's exam paper. So the examiners come together, they discuss what they will, what they won't accept, and they agree before that goes out to everyone. Oh wow, you got an A in biology and you said neuroscience. That's amazing. When will you explain? I'll come back to live when you're explaining. So if I'm short of time, do I make notes on videos or past papers? Past papers. If you're short of time, do as many past papers as you can. When you get stuck, go watch a quick video on it and then try the paper again. Zingy, you're specifically looking at the nucleotides coming together to form the polynucleotide. You really want to look at my DNA replication video. Once you've seen that, you'll be able to hopefully make it make sense. If not, then come back and ask a question. But that's, the video is there for you already. The reason I do these sessions is so that I can answer questions that people are asking, not just go over content. So if you're just looking for content, then please do go watch my videos. But if you've got a specific question on what you're struggling with, then obviously write that and I'll try and counteract it. But didn't answer it. How would you link nucleotide to polynucleotide when teaching? So how would I link it? Nucleotide, the nucleotide is just the DNA nucleotide, or are you talking about the RNA nucleotide? It depends where you want to go. So I can link it with the formation of the phosphodiester bonds by talking about the RNA polymerase or the DNA polymerase, or I can link it by talking about the complementary base pairs. It depends where you want me to go with that. And that's why I'm saying you need to be sure on what you're asking. These sessions are really helpful. You are very welcome. I'm glad you're finding them helpful. Is a neurotransmitter for sympathetic noradrenaline and then parasympathetic acetylcholine? Yes. So the other thing to note, Chidama, is that you need to understand that the somatic nervous system and the autonomic nervous system, the autonomic nervous system is split into the sympathetic and parasympathetic. The somatic nervous system also releases acetylcholine. So make sure you're happy on that one. I'm thinking about getting Snap Revise alongside your videos. Do you think it's worth it? I couldn't say, I haven't seen them, so I can't comment on that. More about the condensation reaction. So Zingy, if you're looking for a condensation reaction is where there is a reaction that is going to bring things together and water is also formed as a product. So if I'm talking about that, you've got two nucleotides that are being brought together through a condensation reaction. What am I getting? A polynucleotide and water as the product. So it depends where you're going. Somatic is the somatic, yes, it is acetylcholine. You're trying to cram it in, you forgot to revise. Oh, bless you. Do you know anything about the CGP books? Yes, I do think the CGP books are good. I have used them myself in the past. Linkage bonds as well. Phosphodiester linkage bonds as well as what, Zingy? Phosphodiester linkage is a different name. It's just a different name for a phosphodiester bond. Different exam boards will call it either linkage or phosphodiester bond. It just depends which exam board you're doing it with. With regards to the CGP guides, going back to that, what you're talking about is finding something that works for you. There's so many different books out there, so many different guides. Yes, all my notes in my videos are mark scheme specific for OCR. Yes. So what does it happen between the nucleotide? Right, hold on. Zingy, I am going to jump over to this because you're asking lots of questions on it and it's really important that you do. So if you are watching on my YouTube channel, you'll be able to see what I draw right now because I've got the screen open and I'm doing it. So if we were looking at the nucleotides, I'm just going to do very basic nucleotide. There's one nucleotide. You know that at the third carbon there is a hydroxyl group. You should know that you also have, if I bring in an adjacent nucleotide, I've got a phosphate group here. Now that phosphate group, what can happen is these two can actually come together, and because we can have a condensation reaction occurring, therefore water is being produced as a product, what's going to happen is these two, hold on one second, fine, I bring another one, are going to join together, and because they're joining together, what we've created is a phosphodiester bond. So here, I'm going to link this. So you can see the difference. So that's my carbon. So it's a pentose sugar, so it's got a fifth carbon, and that's carbon 5. So the carbon 5 is up there. That's why that's called the 5' end. And down here is carbon 3, and that's the 3' end. And therefore, what you are getting is a phosphodiester bond. Two ester bonds. If you were looking at, one second, I'm going to switch screens again. If you were looking at a glycerol molecule, and you look to the glycerol molecule, you know you've got your carbons, you know you've got your hydrogens, you know you've got your hydroxyl groups on my glycerol molecule. And if I now bring in one fatty acid chain, I've got my carboxylic acid group, and then I've got all my carbons, blah, 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 however many carbons I want to add in there. And I've got my hydrogens. So I know that if I'm going to have a condensation reaction occurring, if I remove one water molecule out, so I've got H2O being removed, what's going to happen? What's going to happen is they're going to form a ester bond. So what did I have to form my ester bond? I had a carbon with an oxygen with another carbon. That carbon's double bonded to another oxygen. This one is to the hydrogens. Okay, so my ester bond is being formed here. So that is an ester bond. So what have I got with the phosphodiester bond? Phosphate, I've got a phosphate group. So if I go back to this one here, what have I got? I've got a phosphate there. I've got my, crumbs did not destroy that. Let's try that again. I've got my carbon number 3. So there's a carbon. I've got an oxygen here to the phosphate. I've got an oxygen here to the carbon. So it's a phosphodiester bond because there are 2 ester bonds with a phosphate group. So I hope that helps you, Zingy. Miss, you're so underrated. Just, oh, thank you. It's a funny one. I'm still trying to figure out this whole process of how to actually monetize it enough. I don't get anything on TikTok unless people do give me little gifts, so thank you so much. So far I've been on TikTok for 3 years now, and over the whole time that I've done my lives I've made £1.11, I think it is, but I do appreciate it, so thank you very much. My lady, as you have asked so nicely and you gave me so many roses, I will go through epistasis for you. I was talking about that today actually with my group session. I was touching on it because not everybody has actually covered the topic area, but I think it is important as you're going through patterns of inheritance to really understand the module 2 content along with the module 6 content. So is it the 5th carbon on the sugar and the 3rd carbon from phosphate group? So it's the 5th carbon on the sugar is attached to the phosphate group, and the 3rd carbon of the other sugar is attached to the phosphate group. Zingy, to answer your question. Let's go over the ELISA test. Tills, I'll come back to the ELISA test. So that's AQA. That's not actually in OCR, although different books actually do talk about it, but I will come back to that. That one, if you're given it in the OCR exam questions, you'd be given information. They're not expecting you to memorize the content as being the ELISA, whereas for AQA you are expected to memorize it. Give me 1 second, I'm just trying to bring up my thing on the epistasis. Here we go. Right, let me bring over some slides and hopefully make this make sense for epistasis. Zingy, you are very welcome, but as I would say, go and watch my video on nucleic acids and nucleotides. So it's 2.1.6, because obviously I'm not doing it off the top of my head as I am right now. It is with the detail that you need. There's slides to go alongside that, so please do obviously watch that as well. Right, well, here we go. Epistasis. What is epistasis? So epistasis is the interaction of genes. Biology does cook most people, by the way. It is so much content. I love it. I live and breathe biology, and I absolutely love it. I have a passion for it. I want to teach it as much as I can. But the one thing I would say is there's so much that if you don't keep on top of it as you're going, you are going to struggle, because it gets more and more content heavy. It gets more and more complicated, and especially if you don't have the foundational skills that are needed. Anyway, back to epistasis. So epistasis is this interaction of genes at different loci. Now, when we talk about loci, what we have to remind ourselves is if I have a chromosome and that chromosome is split up into, very basic here, genes. The location of that gene is called the loci. So if I have, let's say, gene A here, I'm going to say this is gene A, and down here is gene B. Now I'm going to put them on the same chromosome just to give this example. However, they may be on, for example, chromosome number 1 and chromosome number 21, and they are going to interact. So I'm just putting it on the same chromosome at the moment. So I'm saying that they're autosomally linked, but what I'm going to explain is what do they do. So epistasis is the interaction of genes at different loci, different locations, and they are going to affect the phenotypic expression, the physical characteristics that we see. You can't see what I'm writing here because I'm on my YouTube channel. So if you jump over to my YouTube channel, you should be able to see what I have there. TikTok doesn't like me saying go over there. However, I can't run both of them at the same time, whereas on my YouTube channel, if you watch my videos, I can monetize it if you watch the ads, sorry, but I can. It's one of the ways in which I now, woohoo, can earn a little bit of money, not much, but a little bit of money on the YouTube channel, which is why I put it there. Anyway, back to this for you. What is epistasis? So if I'm going to start with dominant epistasis. So you should know about dominant allele. So if you've got a dominant allele is going to be one which is going to be expressed, whether you have one or two in your genotype, the physical characteristics from that dominant allele are going to be expressed. So if we have dominant epistasis, what that means is a dominant allele at one locus may mask the phenotype of a second locus. So in this case, if I have gene A, and gene A, I have a dominant A, results in no pigmentation. So here I have my pumpkin. We've just had Halloween. It's really funny. This is actually exactly the time of when I teach this because I use pumpkins all the time. So when you go to the pumpkin patch, for example, you pick your pumpkin, you cut it up, you put it out for Halloween. Why is it you can have the orange pumpkins or the green pumpkins, or in this case, a white pumpkin? So gene A is a dominant A will mean no pigment. Recessive, obviously I need two recessive alleles together. That will therefore result in pigmentation. So if I have got gene B, if I have a dominant allele B, that will give a phenotype of an orange pumpkin. Or if I have recessive to lowercase b's, that will give me a green pumpkin. So what does that actually mean? Well, if I have the dominant allele A, that could result in the production of a protein which is a repressor protein. Now, remember, our genes are sections of DNA that carry the code for proteins. So if I have dominant A, where there's going to be no protein, what can happen is that can result in transcription, copying of the code, translation to the primary sequence of my polypeptide, which could result in a repressor protein. Now, if it's going to result in no pigment and it's a repressor protein, what is going to happen is that repressor protein will bind to the DNA strand. It will block RNA polymerase from being able to bind at the promoter region, therefore preventing transcription from occurring. So if we have a repressor protein, that could actually result in the blocking of the RNA polymerase, therefore no transcription of gene B. So that's one of the ways in which we can prevent, we can interact with another gene at a different location by preventing transcription from occurring. Oh, a second. What it could also be is it could also result in the production of an inhibitor molecule. Now, it will tell you in the exam question, or it will hint to whether it's a repressor protein or whether it's an inhibitory molecule if they want you to give that answer. Or they will accept both answers because they haven't told you what it's going to do. So if I have a gene A, which is dominant epistasis, that allele being present could result in a repressor protein blocking transcription or an inhibitory molecule, which is going to bind to an enzyme that is needed in the pigment production. So it can be either of those two things. We are basically stopping the phenotype from being shown. We're masking that phenotype because of either preventing the transcription occurring of the gene or by inhibiting the production of a protein or the color. So if we're looking at this dominant epistasis then, and we're going to stick with our pumpkin, if I have gene A, no pigment, gene B, orange or green, and I take two white pumpkins, so we know they're white, so I'm going to tell you they're heterozygous. Because they're heterozygous, they are going to have one of each. So I've got a capital A, lowercase a, capital B, lowercase b. So they are heterozygous for both of those genes. Could you have the inhibitor blocking the enzyme RNase polymerase? Yes, so hold on a minute. So could you have the inhibitor blocking the enzyme RNA polymerase? So the inhibitor molecule would be, for example, as talking about a competitive inhibitor or a non-competitive inhibitor. So if we're talking about RNA polymerase, we're talking about an enzyme that has a specific shape, tertiary structure, where it's going to bind and be able to catalyze the reaction. So if we're talking about an inhibitor, they would be looking for you saying whether it's a competitive or non-competitive inhibitor. So if it's blocking the enzyme RNA polymerase, it would therefore need to be a competitive inhibitor for using that terminology that it's blocked it. So the fact that you've asked that question, if they were to ask that sort of question in the exam, that's what they're looking for you to use as your knowledge being brought to the concept. So it's heterozygous because I have a capital and a lowercase. I have a dominant allele and a recessive allele, and both genes are heterozygous. Can I repeat? So the question that Tildes asked was about, one second, let me just find it again. Could you have the inhibitor blocking the enzyme RNA polymerase? If the inhibitor is blocking the enzyme RNA polymerase, they're wanting you to state the fact that it would be a competitive inhibitor. If they are asking about an inhibitor molecule which would bind to the allosteric site, it would change the tertiary structure. That would therefore mean that the active site is not complementary and it can't catalyze the reaction. So it really depends on where the question wants you to go, which part of your knowledge it's assessing as to which answer it's looking for. And again, if you're asking you're answering a question, you can give either of those answers as long as you justify what it is you're stating. If it's an inhibitor molecule and it's blocking it by changing the active site shape, then it's a non-competitive inhibitor. So I can twist my words to make it make sense, but because you said it was blocking and it was an inhibitor, I would have automatically gone that it's a competitive inhibitor. Does that help? Ladies, that answered your question? Good. Going back to this, so I've got heterozygous for both alleles. So if I've got a heterozygous for both alleles, now I said before that they were linked, but remember, most of the times they're not linked. So we're going to pretend that they're not linked. I was just showing that on the diagram. But here, if they're not linked, what's the possible gametes that can happen? Now remember, dihybrid, we've got two alleles 2 alleles, 2 genes. So monohybrid, 2 alleles, 1 gene. Dihybrid, 2 alleles but 2 genes. So here, because I'm doing a dihybrid cross, I am going to take my genotype and I am going to do a reduction division. Meiosis is going to occur, and because of that I'm going to go from a diploid, 2 sets of every chromosome, to a haploid gamete. The haploid gamete having 1 of each. So because of that, I've gone from diploid, 2 A's, 2 B's, to haploid, 1A, 1B. Then what I need to understand is, what's the possible combination? Well, I could have one capital A and one capital B, or I could have had a capital A and a lowercase b, or a lowercase a, capital B, or lowercase a, lowercase b. Well, why am I getting those possible combinations? Because of independent assortment. However they line up at the equator, they are going to independently assort, which could potentially result in all the different possible combinations. So if I've got all the different possible combinations and I place them into my Punnett diagram, I can then fill my Punnett diagram by just coming down and across, down and across. Now, whenever I do this, I always come down, come across, but I keep my letters together just to make it easier on myself. And what I also do is I always make sure that if there is a capital coming either down or across, doesn't matter which way, if there is a capital, I always put the capital first. That therefore means that I can quite easily go through, I quickly do this, and figure out which ones are going to have the dominant. So if they've got the dominant, they're going to have no pigment. No pigment, no pigment, no pigment, no pigment, no pigment, no pigment. So it doesn't matter where I go through, as long as I can see a capital A, anywhere where there's a capital A, I know because of dominant epistasis, there will be no pigment whatsoever. So therefore, the only ones that are going to have pigment are the ones that have the recessive 2 small a's. So 2 small a's and capital B's are orange. 2 small a's heterozygous are still orange. The only one that will be green is the pigment and then the recessive 2 alleles to give me the green pigment. And so therefore I can see that my predicted ratio for my phenotypic ratio is not my usual hetero with hetero should give me a 9:3 to 3:1 if independent assortment has occurred. If independent assortment doesn't occur, therefore something else is at play. In this case, it's epistasis, because I've got 12 white, 3 orange and 1 green predicted, or I could end up with 13 if my recessive color was white. So that would be if it was dominant epistasis, the presence of a dominant allele is resulting in no pigmentation occurring. So I was talking tonight about butterflies, and I was talking about butterfly wings and the different colors on the wings, whether they are brown or orange, or sometimes you end up with the wing color being white, for example. Well, that's because of epistasis. There is an interaction which is resulting in no pigmentation in those areas where there is white. And therefore that white is being reflected, white light is being reflected rather than there being some absorption and some reflection. And so therefore, because of the scattering of the light out from the wings, which is really fascinating, that is why we see the different colors in the butterfly wings, which is where we were talking about that tonight. Now, if we go to recessive epistasis, just to finish off, and I'll finish on that one. Recessive epistasis, still epistasis, we're still talking about interactions. The difference is recessive epistasis, we need 2 recessive alleles. So when we look at, for example, Labradors, so cute, you look at the lab, you look at the color, what you need to understand is That is again because of epistasis. You have gene 1, which gives you either the black lab or the brown chocolate lab. Gene 2 is whether there's pigment that's deposited in the fur, or whether there is no pigmentation deposited in the fur, and that's what gives us the yellow fur. So the golden lab is actually because of epistasis, because it's recessive epistasis, we end up with this yellow lab, and it doesn't matter what we have in gene 1 because the presence of the 2 alleles in gene 2 is masking whatever is present in gene 1. So that's why it's called epistasis, because it's this interaction of the genes from one location to the expression of a gene at another location. So you can quite clearly see I've just done my Punnett diagram. I'm coming down and across, and then I would then identify whether it is a black lab or whether it is a brown lab or whether it is the yellow lab. So when you are looking at the epistasis, the key thing to know is, does the phenotypic ratio follow the 9:3 to 3:1 ratio that we would expect because of independent assortment if we were to cross 2 heterozygous individuals? If it doesn't, if the numbers don't give us a 9:3 to 3:1 ratio, it could either be because of autosomal linkage or it could be because of epistasis. So if they're going to ask about epistasis, they're either going to give you the examples about this gene is dominant or recessive because you can see pigmentation, no pigmentation, color brown, black, or in the last example, green and orange, or it could be down to the predicted ratios. So it really just depends where they want you to go. Right, I'm going to stop there because it's 10 o'clock and I need to go to bed. I will try to come on at 9 o'clock on Wednesday. If I can, I'll try and set up in the house because I'm obviously looking after the girls. You are very welcome. I hope you have found it useful. I hope you do find my videos useful. Please do like, share, tell people about the resources I have out there, because obviously the more people that I can help, the better. And thank you, because I love doing this and I wouldn't be able to do this if it wasn't for you guys joining me. And thank you so much for the roses. I do really appreciate them. Yep, definitely come back. And obviously, if you want to see what I've drawn, what I did, what I discussed, it is actually on my YouTube channel, and you can watch it back at other times. Yes, it is absolutely fine if you start revising from now for year 12. I sometimes run accelerated courses. I sometimes go into some private schools and help them out. And the accelerated course I would run from January through to May and be able to teach the whole of year 12 in that amount of time. Even if you're doing AQA, these resources are helpful. There is a lot of cross overlap that's going on. So do just keep an eye on your specification. The GCSE biology I am launching next week, the free lives that I'm going to do, they're going to be at 6:30 on the Monday. Depending on how much interest I get, depends on whether I will run them every week or not. I was going to be launching it for someone else and they pulled out. Maled, yes, I have covered the whole specification for OCRA on my YouTube channel, and it's in playlist specifically linked to the specification. So if you look at the specification, go to the module 2.1.2, for example, is biological molecules. That will be there for you to look at the resources. Also, all my lives are there that I did last year, so you'll be able to see that too. With regards to the GCSE, combined science is going to be there, so I'll talk about the content which is combined science and also mention when I'm going into the triple biology content. Do I do Edexcel GCSE biology? You can't find anything. The reason you can't find anything for Edexcel GCSE is because there's so few schools that do it. There is, I was looking tonight, 143,000 10,000 students who sat AQA last year. 10,000 sat OCR, and I want to say actually Edexcel was more, 20,000 who sat Edexcel, but there's so few schools that actually run it, so that's why you'll find more resources for AQA. The content is very much the same though with GCSE. It's just about where they put the different topic areas and how the exam questions come out. But yeah, it's one of those things that it's why you'll not find that much out there for your specific exam board, but it doesn't mean that the stuff that's out there isn't relevant to you. Just make sure that you're looking at your specification. That is basically your go-to for everything. Even when I am doing this now, I know OCR inside out, I know AQA GCSE inside out, but I will still have the specification open. I will still double check because there's only so much information I can retain in my brain. I can't find lots of stuff, but thanks for your help anyway. You are very welcome. But honestly, with regards to Edexcel, look at the content. So if it's enzymes, for example, enzymes is enzymes regardless of whether it's Edexcel or whether it is OCR or whether it's AQA. Is it finished for me? I do Salters not. Oh, Ethan, I'm so sorry. That's a rough one. Salters Nuffield exam board is an interesting one, but it is definitely one that I would say is the hardest one for finding the resources for. I tutored a couple of years ago that one, and I'm not going to lie, I didn't enjoy it as much.